A high-altitude gene may unlock the brain's ability to repair itself. Recent research suggests that evolutionary adaptations found in high-altitude animals may hold the key to repairing damaged nerves in the human brain. Scientists have identified a genetic mutation originally evolved to help species such as yaks and Tibetan antelopes survive in low-oxygen environments that also appears to protect and restore the myelin sheath, a critical structure for neural function. This discovery could open new therapeutic pathways for neurological disorders, such as multiple sclerosis, MS, and if you like my videos, subscribe my channel and press the bell icon to turn notifications on so you keep getting all the major advancements in science and technology. And don't forget to like and share this video with your friends and colleagues. The study, published in Cell Press journal Neuron, highlights how naturally occurring genetic variations can reveal biological mechanisms with direct medical relevance. Rather than introducing entirely synthetic treatments, this approach leverages pathways that already exist within the body. The importance of myelin in brain function. The myelin sheath is a lipid-rich insulating layer that surrounds nerve fibers in the central nervous system. Its primary role is to facilitate the rapid transmission of electrical signals between neurons. When myelin is damaged, neural communication becomes inefficient or disrupted entirely, leading to cognitive and motor impairments. In early development, insufficient oxygen supply can impair myelin formation, contributing to neurological conditions such as cerebral palsy. In adults, myelin degradation is a defining feature of multiple sclerosis, in which the immune system mistakenly attacks this protective layer. Age-related reductions in cerebral blood flow can also compromise myelin integrity, contributing to disorders such as vascular dementia. Yet species like yaks and Tibetan antelopes maintain normal brain function under these conditions. Previous studies identified a mutation in a gene known as Retsat in these animals, suggesting it plays a role in their adaptation to hypoxia. To investigate its function, researchers introduced this mutation into laboratory mice and exposed them to low-oxygen conditions comparable to high-altitude environments. The results were striking. Mice carrying the mutation showed improved cognitive performance, including better learning, memory, and social interaction. Importantly, their brains exhibited significantly higher levels of myelin compared to control groups. Enhanced myelin repair and cellular regeneration beyond protection. The mutation also appeared to enhance the brain's ability to repair existing damage. In experimental models simulating conditions similar to multiple sclerosis, mice with the modified Retsat gene demonstrated faster and more complete remyelination. This regenerative effect was linked to an increase in oligodendrocytes, the specialized cells responsible for producing myelin. Not only were these cells more abundant, but they also matured more efficiently, accelerating the restoration of damaged neural pathways. The role of vitamin, a metabolism further investigation, revealed a biochemical mechanism underlying this effect. The Retsat mutation enhances the activity of enzymes involved in vitamin, a metabolism, leading to increased production of a molecule known as ATDR. This metabolite plays a crucial role in promoting the growth and differentiation of oligodendrocytes. When researchers administered ATDR to mice with MS-like symptoms, they observed reduced disease severity and improved motor function. These findings suggest that ATDR or related compounds could serve as a therapeutic agent for conditions involving myelin damage. A new direction for neurological therapies. Current treatments for multiple sclerosis primarily focus on suppressing immune system activity to slow disease progression. While effective to a degree, these approaches do not directly repair damaged myelin. The newly identified pathway offers a complementary strategy, enhancing the brain's intrinsic capacity for regeneration. Because ATDR is derived from vitamin A, a compound naturally present in the human body, this approach may offer advantages in terms of safety and biological compatibility. It represents a shift toward therapies that amplify existing physiological processes rather than introducing entirely external interventions. Translating results from animal models to clinical treatments involves significant challenges, including dosage optimization, delivery mechanisms, and long-term safety evaluation. Nevertheless, this work underscores the value of studying evolutionary biology as a source of medical innovation. Genetic adaptations that enable organisms to survive in extreme environments may reveal solutions to complex human diseases. By linking hypoxia adaptation to neural regeneration, researchers have identified a potential pathway for treating debilitating neurological conditions. If successfully developed into a clinical therapy, this approach could transform the treatment of diseases like multiple sclerosis, not merely slowing their progression, but actively restoring damaged neural function.